In this video, we're going to continue to talk about RNA polymerase. So RNA polymerase cannot read damaged DNA. So if it comes across damaged DNA, it will get stuck during the transcription and just stall right there on the genome. And there's really nothing that can be done about it. It'll just be stuck. However, if DNA or the wrong RNA base is incorporated during transcription, there is something RNAP can do about that. So let's remember RNAP while adding new RNA. The new RNA that's being synthesized will be interacting with DNA and making a helix. Well, if the wrong base is added or DNA is added, this helix will be distorted, and this distortation will cause the RNAP to stall. And in fact, it can't really move forward when this helix is distorted. It sits there. But one thing that RNAP can do is that it can go in reverse. And by that, I mean the I minus 1, the nucleotide that was there, goes to the I plus 1. And the I plus 1 will now go into a separate channel that we haven't talked about before. This is called the proofreading site. And this backtracking is a way for RNAP to do its own types of corrections. So what will happen is that if RNAP gets stalled, it will no longer move forward, and now moving backwards is preferred. And it can't move forward until the wrong base is eliminated. So there are other proteins that are associated with this. So GREA and GREB will help hydrolyze this in E. coli, while TF2S will hydrolyze this base in RNAP2. And so this cut has to be done at the active site. So it cuts between the I minus 1 and the I plus 1 region. We cut the phosphodiester bond. We remove the nucleotide, and then we can continue to move forward. Now, let's talk about how we start RNAP in eukaryotes, and that's with a promoter region. Now, in DNA synthesis, DNA polymerase, we talked about the sigma unit, which binds the promoter and then falls off. RNAP does not have the equivalent of a sigma. But there are other proteins that will bind the promoter region in the genome first and then recruit RNAP to that start site. So it's similar to a sigma in DNA polymerase, but it doesn't actually bind to the RNAP complex to begin with like sigma did. Now, the promoter regions for RNAP are species specific. This is actually not the case for RNAP2 and RNAP3, but that is the case for RNAP1. In the core promoter element for RNAP1 is between minus 31 and plus 6. So the promoter for RNAP1 is actually in the region that gets transcribed. Now, you need a lot of elements for eukaryotic initiation. It's a lot more complex than prokaryotic. And so you need an upstream promoter element that's roughly minus 187 to minus 107, which will have a lot of GC nucleotides in there. And this upstream element has transcription factors, other proteins that bind there, and then again will recruit RNAP1 to that part of the genome. Now let's take a look at RNAP2's promoter region. And like I just said, it's way more complex than the prokaryotic version. First off, different types of genes will have different upstream transcription start sites. So if you're a housekeeping gene, that is a gene that always needs to be active, you're going to have a lot of GC-rich regions. These are called CpG islands. So the P just means a phosphate bond, so C phosphate bond G over and over and over again. However, if you're a gene that doesn't need to be on all the time and is only selectively transcribed, you're not going to have this GC island, or CpG island rather. So CPG islands can tell us which genes are active all the time and which ones are not. You also need some core promoter regions that are roughly 40 to 50 nucleotides, and they can include the start site. So one or more of these elements help direct transcription initiation. And probably the best one you know about is, and the one that scientists know about is the Tata box. So this is minus 31 to minus 25 upstream, and it has a lot of ATs. That's why it's called Tata. Now, what's interesting though is that even though this is the best known promoter element, best known core promoter, 2/3 of the genes that encode proteins don't actually have this. So if you don't have a Tata box, you're going to have something else. You're going to have this motif 10 element or this DPE. However, you're not going to have all of these elements in any single gene. If you don't have one, like the Tata box, you're going to have something else. We also have this initiator element, which is in the transcribed gene again, minus 2 to plus 5. And roughly 50% of our genes will have this initiator sequence. And something not shown here, another type of core promoter would be the Kata box. And this is minus 70 to minus 90. Now, if a gene has the Kata box, this actually will slow down gene transcription. So it makes RNAP2 less likely to bond. So this is a way to regulate the rate at which gene transcription occurs right in the genome itself. We don't need a protein to do this. The genome is directing how often it will be transcribed itself. And we also see that with the CPG islands. Last thing we're going to talk about in this video are enhancers. And what's cool about enhancers is that it doesn't seem to really matter where you place them or what orientation they're in. For example, if you take an enhancer and flip the 5' to 3' or switch it from the sense to the antisense strand, it still works. If you move them around, it still works. So it doesn't have a position or orientation factor like our core promoters did, where we talked about the Tata box having to be in a certain position. Now these enhancers are in cellular genes and also eukaryotic viruses, and that's where we first found them, is from studying viruses. So if a gene has an enhancer, these are required for the activation of the promoter sequences. And these enhancers are bound by specific transcription factors. And these transcription factors will allow RNAP2 to bind the corresponding promoter. Like I said, position doesn't matter. So you could be far away from your actual promoter where this enhancer is. And since you can be far away in the genome, this must mean that the enhancer and promoter must loop around. The DNA is looping on itself so that the transcription factor can contact the enhancer and the RNAP2 at the promoter. And so that's something to keep in mind. Even though something could be 1,000, 2,000 base pairs away in sequence, DNA is like a wet noodle. It's like a rope. It can bend back on itself. So even though sequence-wise they're far away, inside the cell, distant-wise, they could actually be super close to each other. And these enhancers help to mediate the selective gene expression in eukaryotes. Now, RNAP3 promoters, didn't talk much about RNAP3 yet. Their promoters are actually located entirely in the transcribed region, which is really odd to think, but the way to get RNAP3 genes transcribed is that they have to be in the transcribed region to begin with. And that's it for this video. I will see you in the next video.